This is me Nafisa and today we are going to learn about classification of bacteria. Classification of bacteria is on the basis of their pathogenicity. Those bacteria which are capable of causing diseases or infections are known as pathogenic bacteria or harmful bacteria or bad bacteria. Example, Salmonella typhi causes typhoid in human beings. Bacteria which do not cause infections are known as non-pathogenic bacteria. Some bacteria are useful and are known as good bacteria. Example, lactobacillus or lactic acid bacillus present in GI tract of humans. Bacteria are so small that they can be seen only with the help of a microscope and cannot be seen with the naked eye. Naked eye can see up to 200 microns. Length of bacteria generally is 3 microns to 5 microns and diameter generally is 0.2 microns to 1.5 microns. Therefore, bacteria can be seen only under microscope. When viewed under the microscope, bacteria have different shapes or morphology. Second classification of bacteria is on the basis of shape or morphology as coccus. These bacteria are spherical or oval-shaped cells. Coccus means berry. Bacilli are rod-shaped bacteria. Vibrios are comma-shaped and named as Vibrio as they have vibratory motility. Spirilla are rigid spiral forms. Spirochetes are bacteria with flexible spiral shape. Mycoplasma do not have a cell wall, therefore do not have a stable morphology. Actinomycetes are branching filamentous bacteria. Actis means rays and mycus means fungus. As actinomycetes looks like the rays of the sun, therefore the name actinomycetes. These bacteria are further classified on the basis of arrangement. That is, cocci are classified further on basis of arrangement as coccus, single-celled, diplococci arranged in pairs, tetrads arranged in group of 4s, sarcina arranged in group of 8s, streptococci arranged in chains, staphylococci arranged in grapes-like cluster or in bunches. Bacilli are classified further on basis of arrangement as diplobacilli arranged in pairs, streptobacilli, bacilli arranged in chains, Corynebacterium arranged at angles forming Chinese letter pattern, Cocobacilli. A Cocobacillus is a type of bacterium with a shape intermediate between cocci and bacilli. That is, they have very short rods which may be mistaken for cocci. Classification of bacteria is on the basis of differential staining. Differential staining ensures different colors for different bacteria. Two most widely used differential stains are Gram stain and acid-fast stain. Gram stain invented by Christian Gram in 1884, hence the name Gram stain. This staining differentiates bacteria into two groups. If bacteria retains primary stain, that is, if they appear violet under the microscope, they are called as gram-positive bacteria. And if bacteria take up counter stain, that is, appear red when seen under the microscope, they are called as gram-negative bacteria. The second staining is acid-fast staining. Commonly used is Ziel-Nielsen's stain. Acid-fast bacteria appears red in color when seen under microscope. Example, Mycobacterium tuberculosis. Classification of bacteria on the basis of gaseous requirement or classification of bacteria on the basis of growth requirements. Obligate aerobes. Those bacteria that grow only in the presence of oxygen and cannot grow in the absence of oxygen. Facultative anaerobes. Those bacteria that are able to grow either with or without free oxygen. Microaerophiles grow best in presence of lower levels of oxygen than that present in the atmosphere. Obligate anaerobes, those bacteria that can grow only in the absence of oxygen. In fact, these bacteria will die if there is oxygen. Capnophiles, those bacteria that require carbon dioxide for growth. They are carbon dioxide loving Bacteria are also classified on basis of their locomotory organ, that is flagella. Bacteria which possess one or more flagella are called as motile bacteria, and non-motile bacteria are without flagella. Non-motile bacteria without flagella are called as atrichous bacteria. Motile bacteria are further classified as monotrichous bacteria, bacteria having single flagellum in one end of the cell. Amphitrichous bacteria are the bacteria having single flagellum at both ends of the cell. Lophotrichous bacteria are bacteria having bundle of flagella or tufts of flagella at one or both ends of the cell. Peritrichous bacteria are bacteria having flagella all over the cell surface. Classification of bacteria on the basis of spore, that is, spore-forming bacteria are those bacteria that produce spore during unfavorable condition, and the non-sporing bacteria are those bacteria which do not produce spore. Spore-forming bacteria are further divided into two groups as endospore-forming bacteria, that is, they produce spores within the bacterial cell, and exospore-forming bacteria, that is, produce spores outside the cell. Classification of bacteria on the basis of mode of nutrition. Those bacteria which gains energy from sunlight are known as phototrophs. On the basis of source of electron, phototrophs are further divided into two groups. Phototropic bacteria which uses inorganic compounds as electron source are called as photolithotrophs. Phototropic bacteria which uses organic compounds as electron source are called as photoorganotrophs. Those bacteria which gains energy from chemical reactions are called as chemotrophs. They cannot carry out photosynthesis. On the basis of source of electron, chemotrophs are further divided into two groups. Chemotropic bacteria uses inorganic compounds as electron source are called as chemolithotrophs. Chemotropic bacteria using organic compounds as source of electron are called as chemoorganotrophs. Bacteria which uses carbon dioxide as sole source of carbon to prepare its own food are called as autotrophs. They are capable of independent existence. Those bacteria which are unable to grow with carbon dioxide, these bacteria are dependent on preformed organic compounds are called as heterotrophs. Most of the human pathogenic bacteria are heterotrophic in nature. Classification of bacteria on the basis of temperature requirements. Microbes can be classified according to the range of temperatures at which they can grow. Temperature at which bacterial growth occurs best is the optimum growth temperature for the organism. The lowest temperature at which the organism can survive and replicate is its minimum growth temperature. The highest temperature at which growth can occur is its maximum growth temperature. Philes. Suffix phile means loving. Psychrophiles literally means cold loving, are microorganisms that can grow at 0 degrees Celsius, having an optimum growth temperature of 15 degrees Celsius and a maximum growth temperature of less than 20 degrees Celsius. Mesophiles are microorganisms which grow at moderate temperatures between 20 degrees Celsius and 45 degrees Celsius and with an optimum growth temperature in the range of 30 to 39 degrees Celsius. Most human pathogens that are dangerous grows best at the normal human body temperature 37 degrees Celsius. Organisms that can grow at optimum temperatures of 50 degrees Celsius to a maximum of 80 degrees Celsius are called as thermophiles, literally meaning heat-loving. They do not multiply at room temperature. Thermophiles are widely distributed in hot springs. Hyperthermophiles literally means extremely heat-loving, refers to a microorganism that has an optimum growth temperature above 80 degrees Celsius. Classification of bacteria on the basis of optimum pH for growth. Acidophiles are those bacteria that grows best at acidic pH. Alkaliphiles are those bacteria that grows best at alkaline pH. Nutrophiles are those bacteria that grows best at neutral pH. Classification of bacteria on the basis of salt requirement. Halophiles are those bacteria that require sodium chloride or salt for growth. The name halophiles comes from the Greek word salt-loving. Halotolerant organisms do not require salt but can grow under saline conditions.